Hello friends, in the previous videos, we have seen the overview of eukaryotic DNA replication. We also compared prokaryotic and eukaryotic DNA replication. You can refer my videos, the link is given in the description box. In this video, we are going to discuss about eukaryotic DNA replication. We will discuss all the three steps: initiation, elongation and termination of replication in detail. Now let us see the initiation step of eukaryotic DNA replication. Mechanism of eukaryotic DNA replication is similar as prokaryotic DNA replication, but certain differences are there. This we have also discussed earlier. First is larger size of DNA compared to prokaryotes. Hence, multiple origin of replication is present in order to finish the replication on time. Linear DNA. Hence, end structures are present called telomere. Therefore, end replication is required, which is done by telomerase. This we will discuss in detail in separate video. DNA packaging distinctively involves histone proteins. So we already know that in eukaryotes, DNA is a coiled structure. This we will discuss in detail in separate video. Because of these complications, DNA replication is complicated and it involves several enzymes and other proteins. We already know that DNA replication has three steps: initiation, elongation and termination. Here we will discuss about initiation step. Initiation, it is to start or to initiate. It needs to be regulated because of multiple origin of replication. Hence, replication is initiated at many sites simultaneously. So, for each part of genome to be replicated exactly once per cell cycle, tight control is required. If it is unregulated or there is any error in the regulation, then it leads to over-replication that causes DNA damage and genomic instability. This may be due to the repeated initiation events at the same origin. To prevent this, replication initiation is divided into two steps. First is licensing, loading of inactive precursors of MCM2 to 7 helicase. Second is firing, that is activation of the replicative helicase, and these occur in different phases of cell cycle. Licensing occurs in G1 phase and firing occurs in S phase. These two steps are interconnected. Replication begins at origin, which has conserved DNA sequences. In yeast, it is known as ARS, autonomously replicating sequence. Now let us see different events that takes place during the initiation in detail. ORC, that is origin recognition complex, binds to the origin of replication. This then recruits CDC6 protein, cell division cycle 6 protein. After that, CDT1 and MCM227, which is hexamer, bind the ORC CDC6 complex. CDT1 is chromatin licensing and DNA replication factor 1. This CDT1 stabilizes single MCM227 hexamer and loads onto the chromosome. Hence, what we say that MCM227 is loaded. This MCM227 forms the core of the DNA replicative helicase. Both ORC and CDC6 are bound to ATP. So, loading of MCM227 triggers ATP hydrolysis, and this releases CDC6 and CDT1. Now what happens? Loading of another MCM27 takes place. Same steps are repeated and CDC6, ORC and CDT1 are released. Now two MCM27 hexamer are present. This forms double hexamer. This double hexamer encircles double-stranded DNA. Thus pre-replication complex is formed. So this is the origin of replication. ORC comes and binds to it. Then it recruits CDC6 protein. now CDT1 and MCM27 comes and bind to this ORC CDC6 complex. MCM27 is loaded by CDT1. And this loading of MCM227 triggers ATP hydrolysis, as a result of which CDC6 and CDT1 is released. Now there are ORC and MCM227 present. Again the process is repeated. Again ORC recruits another CDC6 protein, and then CDT1 and MCM comes and binds to this ORC CDC6 complex. Same process is repeated. MCM227 is loaded and CDC6, ORC and CDT1 is released. Now this is double hexamer present at the origin, and these encircles double-stranded DNA. This is known as pre-replication complex. So all the above mentioned events takes place in the licensing step of the initiation, and these events occurs in the G1 phase of the cell cycle. As the transition of cell cycle takes place from the G1 to S phase, level of kinase protein DDK, that is DBF4 dependent kinase, and SCDK, that is S phase cyclin dependent kinase, increases. This DDK then phosphorylates MCM227 complex, which then allows recruitment of SLD3 and CDC45 protein. Now this SCDK promotes the formation of preloading complex. This preloading complex is composed of SLD2, DPB11, GINS and DNA polymerase epsilon. Here CDC45 and GINS, these are helicase co-activator. This is the first helicase co-activator and this is the second helicase co-activator. Now this SCDK, that is S phase cyclin dependent kinase, phosphorylates SLD2 and SLD3 protein. This allows preloading complex to attach. This delivers GINS in combination with DNA polymerase epsilon to nascent replication complex. Now GINS associates with CDC45 and MCM227 to form CMG complex. CDC45, MCM227 and GINS complex. This is functional replicative helicase. SLD2, SLD3 and DPB11 dissociates. Hence, double hexamer, which we discussed earlier, is now converted into 2-CMG complex. This double hexamer encircles double-stranded DNA, and as it is converted into 2-CMG complex, then it functions as the helicase. That means it is functional replicative helicase, as a result of which unwinding of the double strand of the DNA takes place by breaking the hydrogen bond between the base pairs. Hence, this CMG complex now encircles single-stranded DNA. And through GINS, DNA polymerase epsilon interacts directly with CMG complex. MCM10, which is associated with the replication origin, then facilitates the recruitment of RPA, which binds to the single strand of DNA and stabilizes it and prevents its re-annealing or rewinding. It also facilitates the recruitment of DNA polymerase alpha with the help of CTF4. This DNA polymerase alpha synthesizes RNA primer less than 10 nucleotides long, which is then followed by the synthesis of DNA stretch less than 15 nucleotides long. So this DNA polymerase alpha has 4 subunits. 2 subunits function as the primase and synthesizes RNA primer, and the other 2 subunits function as the DNA polymerase and synthesizes DNA stretch less than 15 nucleotide long. Then DNA polymerase delta is loaded. Leading and lagging strand synthesis takes place by DNA polymerase epsilon and DNA polymerase delta. So DNA polymerase epsilon synthesizes leading strand and DNA polymerase delta synthesizes lagging strand, but it can be vice versa. because it is not fully clear and research is still going on. So when the transition of the cell cycle takes place from the G1 to the S phase, then DDK, that is DB4 dependent kinase protein, and SCDK, S phase cyclin dependent kinase protein, their level increases. DDK then phosphorylates MCM227 complex or the hexamer. This phosphorylation allows recruitment of CDC45 and SLD3. The CDC45 is helicase co-activator. SCDK promotes formation of preloading complex that includes SLD2, DPB11, GINS and DNA polymerase epsilon. Now what happens? This SCDK phosphorylates SLD2 and SLD3. This phosphorylation allows the attachment of preloading complex, as a result of which now GINS along with DNA polymerase epsilon comes in contact with CDC45 and MCM227 hexamer and forms CMG complex, which means CDC45, MCM227 and GINS. This is the functional replicative helicase. Also DPB11, SLD2 and SLD3 dissociates or these are released. So as the two CMG complex is formed, the double hexamer, which encircles double-stranded DNA, is now converted into 2 CMG complex. Since it is functional replicative helicase, hence unwinding of the 2 strands of DNA takes place by breaking the hydrogen bond between the bases. Now CMG complex encircles single-stranded DNA. Also MCM10 associates with replication origin, then it recruits RPA and DNA polymerase alpha with the help of CTF4. So here MCM associates with replication origin, then it recruits RPA, which is single strand binding protein which binds with the single strand of the DNA and stabilizes it and prevents its re-annealing or rewinding. It also recruits DNA polymerase alpha with the help of CTF4, and this DNA polymerase alpha has 4 subunits to function as the RNA primer and to function as the DNA polymerase. Hence, this DNA polymerase alpha synthesizes RNA primer less than 10 nucleotides long, and then it is followed by the synthesis of small stretch of DNA that is less than 15 nucleotides long. Then DNA polymerase delta is loaded and synthesis of leading strand and lagging strand takes place. This we will discuss in detail in elongation step of eukaryotic DNA replication. Now let us discuss about elongation step of eukaryotic DNA replication. Elongation is the synthesis of 2 new daughter strands complementary to the template strands. It is the second step of eukaryotic DNA replication. Now what is elongation or what happens during elongation? Let us see the overview or let us summarize it. Range of proteins bound during initiation, and these then unwound DNA at the origin. So as we have seen in the initiation step, that number of proteins comes and bind to the origin of replication. This results in the unwinding of the DNA. This takes place in initiation. Then what happens? DNA polymerases are loaded. These then extend daughter strands with the help of certain other proteins. So in this video, in the elongation step, we will discuss about these in detail, that how the polymerases are loaded and how the extension of the daughter strands takes place or how the elongation occurs, and what are the other proteins that are involved in this step. Some of the facts of DNA elongation. Direction of elongation, or the direction in which the new strand is synthesized, is always in the 5' to 3' direction. Primer is required. Major replicative or the main replicative enzyme that are involved in the elongation cannot initiate the synthesis of new strand. They always require 3' OH group for the extension or elongation of the new strand. Hence, primer is required. Addition of new nucleotides require energy. So, in order to extend or elongate the new strand, nucleotides are added which are complementary to the template strand. And during the addition of nucleotides, energy is required in the form of ATP. Elongation is semi-discontinuous. As DNA is antiparallel and the elongation or the synthesis of new strand always takes place in the 5' to 3' direction, and hence one of the daughter strand is synthesized continuously, which is known as leading strand, and the other strand is synthesized discontinuously, known as the lagging strand. Hence, DNA replication is semi-discontinuous in nature. Proofreading also occurs in elongation. Sometimes wrong base is incorporated. DNA polymerase, when recognize this wrong base, then it moves backward, remove or excise this base, and this is followed by the reinsertion of correct base. This is known as proofreading. Now let us discuss enzymes and proteins involved in elongation of eukaryotic DNA replication. First is DNA polymerase alpha. This DNA polymerase alpha synthesizes RNA primer, followed by the synthesis of a stretch of DNA. Primer is synthesized once on the leading strand and at the start of each Okazaki fragment on lagging strand. DNA polymerase delta. It is responsible for the synthesis of lagging strand. It is highly processive due to the sliding clamp pCNA. It has 3'-5' exonuclease activity, hence it shows proofreading activity. Now DNA polymerase epsilon, it synthesizes leading strand. It also has high processivity due to pCNA. It also shows 3'-5' exonuclease activity, or it has proofreading activity. Then CTF4. This is required for the recruitment of DNA polymerase alpha. pCNA, proliferating cell nuclear antigen, which is sliding clamp. It increases the processivity of DNA polymerase. RFC, it is clamp loader, it loads pCNA. It is responsible for polymerase switching, that is replacement of DNA polymerase alpha by main replicative DNA polymerase, that is DNA polymerase epsilon and DNA polymerase delta. Chain elongation reaction, it is same as prokaryotes. So as the nucleotides are added during the synthesis of the new strand or the extension of the new strands, then certain reaction takes place. It is same as in the prokaryotes. This we have already discussed in elongation step of the prokaryotic DNA replication in detail. You can refer my video, the link is given in the description box. Now to understand the elongation step, let us start with some of the events that takes place during the initiation. This we have also discussed in the previous video. So we have seen that during the initiation, when CMG complex is formed, the CMG complex is functional replicative helicase. Now what happens? Parental DNA unwinds at the origin, that is the two strands of the DNA separates. This results in single strand of DNA at the origin. Now the MCM10 is recruited or it attaches to the CMG complex. This MCM10 then facilitates the recruitment of RPA, replication protein A, which is single strand binding protein which binds to the single strand of the DNA and stabilizes it, and also facilitate the recruitment of DNA polymerase alpha with the help of CTF4 protein. DNA polymerase alpha then synthesize RNA primer followed by a stretch of DNA. So what happens? CMG complex is formed, which consists of CDC45, MCM227 and GINS. This is functional replicative helicase. This causes the unwinding of the DNA at the origin. Now the two strands of the DNA unwinds and this results in the single strand of the DNA. MCM10 attaches to the CMG complex. It facilitates the recruitment of RPA. which is single strand binding protein, and it stabilizes the single strand of the DNA, which results due to the unwinding of the DNA. Also, DNA polymerase alpha is recruited. This DNA polymerase alpha synthesizes stretch of RNA primer, which is in orange color, followed by synthesis of a stretch of DNA, which is in green color. Then RFC, replication factor C, which is clamp loader, causes dissociation of DNA polymerase alpha. Because DNA polymerase alpha is not major replicative polymerase, as it has low processivity and it also lacks the 3-5- exonuclease activity. Hence, the DNA strand that is synthesized by DNA polymerase alpha is not accurate. And after the dissociation of this DNA polymerase alpha, this RFC also causes the assembly of pCNA, which is proliferating cell nuclear antigen, which is sliding clamp, same as the beta clamp, which holds DNA polymerase delta and DNA polymerase epsilon to the template strand when loaded. So, this is responsible for the processivity of DNA polymerase delta and DNA polymerase epsilon, which are the major replicative DNA polymerase. So, this process in which DNA polymerase alpha is replaced by the major replicative polymerase, DNA polymerase delta and DNA polymerase epsilon, is known as polymerase switching. Then the DNA polymerase epsilon synthesize leading strand, also DNA polymerase delta synthesize lagging strand. DNA polymerase epsilon is already recruited to the CMG complex, but it cannot initiate the DNA synthesis as it requires 3-OH group for the extension of the new strand, which is provided by the primer. Hence, once the DNA polymerase alpha synthesizes the primer, then only the main replicative enzyme DNA polymerase epsilon and also DNA polymerase delta is loaded. Primer is synthesized by DNA polymerase alpha once for the leading strand and at the start of each Okazaki fragment for the lagging strand. DNA polymerase alpha, because of its slow processivity, and it also lacks the 3-5- exonuclease activity. Hence, the DNA that is synthesized cannot be accurate. It is not the main replicative enzyme. So, once the primer is synthesized, this DNA polymerase alpha is dissociated and the main replicative DNA polymerase, that is DNA polymerase epsilon and DNA polymerase delta, is loaded. So, what happens? This RFC, which is replication factor C, which is also the clamp loader, causes the dissociation of the DNA polymerase alpha and assembly of pCNA, both to the template of the lagging strand and to the template of the leading strand. This pCNA holds DNA polymerase epsilon and DNA polymerase delta, and it is responsible for the high processivity of this main replicative DNA polymerase. So, the process of replacement of DNA polymerase alpha by the main replicative DNA polymerase, that is DNA polymerase epsilon and DNA polymerase delta, is known as polymerase switching. So, this DNA polymerase epsilon synthesizes the leading strand, DNA polymerase delta synthesizes the lagging strand. This lagging strand is synthesized in the form of the small fragment. So, DNA polymerase delta synthesizes a stretch of the DNA, then it gets dissociated. Then it is again loaded to the next primer and the process is repeated again and again till the completion of the synthesis of lagging strand. So, what we have discussed, that one of the strand is synthesized continuously and the other strand is synthesized discontinuously in the form of the Okazaki fragment. For the synthesis of this continuous strand, known as the leading strand, DNA polymerase epsilon is loaded once and then it continuously synthesizes the new strand. But for the synthesis of the lagging strand, DNA polymerase delta is loaded, then it synthesizes a small stretch of the DNA. Then it gets dissociated and is loaded to the next primer. Then the process is repeated. This process is repeated again and again till the completion of the synthesis of the new strand. So why this is synthesized continuously and this is synthesized discontinuously? This is because the new strand is synthesized always in the direction of 5' to 3'. Since this strand, the 5' to 3' is in this direction and hence it is synthesized in the direction same as the movement of the replication fork. As the replication fork moves in this direction, the double helix of the DNA separates and hence the single strand template for the synthesis of this strand is continuously available. Hence it is synthesized continuously. But if we talk about this strand, here 5' to 3' is in this direction, which is opposite to the direction of the movement of the replication fork. As a result of which, small stretch of the single strand template is available, small stretch of the DNA is synthesized. Then as the replication fork moves or the double helix of the DNA separates, again a small stretch of the single strand template is available. Then again small stretch of the new strand is synthesized. In this way, this process is repeated again and again till the completion of the synthesis of new strands. Hence, it is synthesized in the form of the small fragments known as the Okazaki fragment. Also, this strand is known as the leading strand and this strand is known as the lagging strand because synthesis of this strand starts early, and when a certain stretch of this strand is synthesized, then only a stretch of single-stranded template is available for the synthesis of this strand. Hence, the synthesis of this strand starts late compared to this strand. So, this strand lags behind this strand. So, it is known as the leading strand and it is known as the lagging strand. Now, let us discuss termination step of eukaryotic DNA replication. So, what we know that after elongation, termination takes place and it gives two separate copies of DNA. And as the two separate copies of DNA are obtained, this marks the completion of DNA replication or the end of the DNA replication. In this process or in termination, DNA synthesis is completed, the replication machinery is disassembled and daughter molecules are resolved. Now the termination sites. It is determined by position of replication initiation sites. We know that there are multiple origin of replication and hence multiple replication bubbles are formed, and the site where the adjacent replication bubble converges or meets or the adjacent replication fork meets is the termination site. But in case of the prokaryotes, there is specific locus known as the ter site where the termination takes place or the two replication fork meet, and this is diametrically opposite to the origin of replication. As in prokaryotes, DNA molecule is circular, but in case of eukaryotes it is linear. So this is the linear DNA molecule. There are multiple origin of replication, and as the two strands separate at the origin of replication, then the multiple replication bubbles are formed. Strands in red color are the parental strand or the template strand, and those in blue color are daughter strands. Each replication bubble has two replication fork which move away from origin in opposite direction to each other. Hence, it is bidirectional. When the adjacent replication bubble, or in other words, when the adjacent replication fork meets or converges, then the termination of DNA replication takes place. Now let us understand this with the help of diagram. We have taken two origin of replication, and when the strands separate, two replication bubbles are formed. Each replication bubble has two replication forks. As the helicase proceeds, two strands separate and hence the replication fork also moves or proceeds, and both the replication fork of the replication bubble move away from origin in opposite direction. So replication fork of this replication bubble, one will move in this direction and one will move in this direction. Similarly, if we talk about this replication bubble, the replication fork will move in this direction and this replication fork will move in this direction, that is opposite to each other. when the replication fork of adjacent replication bubble meets, or when the adjacent replication bubble converges, then the termination of DNA replication takes place. So as the helicase move and the DNA strand separates, then the leading strand of replication bubble 1 reaches the lagging strand of the replication bubble 2. And in case of the lagging strand, as the lagging strand is synthesized in the form of the Okazaki fragment, so this Okazaki fragment will reach the 5' end of the previous Okazaki fragment. This is the 5' end and this is the 3' end. In this way, the process continues. Similar will be the case, the leading strand of this replication bubble reaches the lagging strand of the first replication bubble. So what is actually happening? The replication fork of this replication bubble is moving in this direction. And for this replication bubble, it is moving in this direction. So this stretch of the DNA between the adjacent replication bubble 1 and 2 is also replicated. And when the DNA polymerase reaches a section of the DNA template that has already been replicated in case of this replication bubble and also in case of this replication bubble, then the DNA polymerase halts. That means this entire section of the template is replicated, and thus the termination of the replication takes place. Similar will be the case for the other adjacent replication bubble, as the eukaryotic linear DNA has multiple origin of replication and hence multiple replication bubbles are formed. Now let us discuss the events that takes place during termination. As discussed earlier, adjacent DNA replication forks or replication bubbles converge or move towards each other during termination. This results in unwinding of stretch of DNA between two adjacent forks. So as the two adjacent replication fork converge or move towards each other, the stretch of the DNA which is present between the two adjacent forks unwind. CMG complexes along with associated proteins approaches each other. So CMG complex, which is functional replicative helicase, results in the unwinding of the stretch of the DNA. Hence, as the replication fork proceeds, CMG complexes, which is associated with other proteins, approaches each other, or these proceed towards each other. Then these CMG complex bypass each other. This is because synthesis of lagging strand is slower compared to leading strand, and hence certain stretch of the DNA is unreplicated, as a result of which CMG complex along with other proteins including the DNA polymerase bypass each other in order to replicate the unreplicated stretch of the DNA. Then these are associated onto the double-stranded DNA. This is followed by the processing of Okazaki fragments, means removal of the RNA primer and replacement it with the stretch of the DNA. This we will discuss in detail in separate video, removal and replacement of RNA primer. This is followed by recruitment of SCF E3 ubiquitin ligase complex. This SCF catalyzes the ubiquitination of MCM227 on the 7th protein. So we know that MCM227 is hexamer. It consists of 6 monomeric subunits numbered from 2 to 7. So this SCF catalyzes the ubiquitination of 7th protein of MCM227 or MCM hexamer. So normally what happens? Ubiquitinated protein is degraded by proteasome, which is known as proteasomal degradation. But here this ubiquitinated MCM227 is recognized by P97, also known as valucin containing protein, in short VCP. This is segregase protein or segregase enzyme, which causes the disassembly of CMG complex along with its cofactors and other proteins. Hence, these are removed or segregated from the DNA strand. Then any catenanes in the daughter DNA strands are removed by topoisomerase 2. This we have already discussed in topoisomerase video. You can refer my video. The link is given in the description box. Therefore, two newly synthesized strands are segregated. Hence, two copies of DNA are obtained. Now, let us understand this with the help of the following diagram. Strands in red color are parental strand and those in blue color are the newly synthesized strand. Stretches that is shown by orange color are RNA primer. This is the CMG complex. During the termination, what happens? The two replication fork move towards each other. Arrow represents the direction of the movement of the replication fork. That is, these converge each other. As a result of which CMG complex also along with other proteins converge or move towards each other. CMG complex, which is functional replicative helicase, causes the unwinding of the double-stranded DNA. It is also associated with other proteins including the DNA polymerase. Hence, new strand is synthesized. These encounter each other. Then these bypass. Here, for convenience, we are just representing the MCM 2 to 7, but it is actually CMG complex. So these bypass each other. This is because leading strand is synthesized faster and lagging strand is synthesized slower. Hence, these move away from each other in order to synthesize the stretch of the template that is unreplicated. Once the synthesis of new strand is complete, then the processing of Okazaki fragment takes place. That is, RNA primer is removed and it is replaced with stretch of DNA. This we will discuss in detail in separate video on primer removal. Then recruitment of SCFE3 ubiquitin ligase complex takes place. This is SCF. This causes the ubiquitination of MCM2 to 7 at the 7th protein. Now this ubiquitinated MCM2 to 7 is recognized by P97, valusin containing protein, which is the segregase enzyme or segregase protein, and this causes the segregation or disassembly of CMG complex along with its other cofactors and proteins. This is followed by the decatenation of the newly synthesized strand. That is, if any catenanes are present between the newly synthesized strand, then it is removed by topoisomerase 2 and hence the two newly synthesized strands are separated. And this gives two double-stranded DNA molecules. This is all for today's video. In the next video, we will discuss about primer removal and its replacement by DNA. So stay tuned. If you like this video, please hit the like button, share it and subscribe my channel. Thank you.